Good afternoon, folks. Mr. Parkinson here. Here's my hands. Now, this is my first ever go at this, so let's see how this works. I'm going to talk to you this afternoon about gene expression, and I'm going to use a couple of examples of how that goes on. Within the specification, the example that you have to use is called the lac operon. Okay. Now, what gene expression is about is the idea that some cells can express a gene to produce a certain protein and other cells don't. And sometimes even a cell will produce a protein sometimes and other times it won't. Now, this makes complete sense because you don't always want to produce that protein, and certainly if you're a skin cell, for instance, you don't want to be producing insulin, and similarly if you're a cell in the pancreas, you don't want to be producing collagen. So we're going to start with a prokaryotic situation, which is somewhat different to the eukaryotic situation. And in the prokaryotic situation, we have an example of what's called the lac operon. Now, this is basically the production of enzyme called beta-galactosidase, easy for me to say. And this is about the idea that if you put a bacteria in lactose, it will start to produce this lactase enzyme to break it down. And if you look at activity 3.14, that's the practical that you may or may not have done in class, which will show you this. But if you put the bacteria in a solution not containing lactase, then it won't produce the relevant enzyme. So here it goes. Now, this blue piece here is a piece of DNA. Where I'm pointing here, this is called the operator gene, and this here is called the promoter region, this green piece here. Now, on the promoter region, this is where the transcription takes place. So if I put the RNA polymerase on there, I would get transcription, and therefore translation of that gene to produce the lactase enzyme. But I don't want that all the time. So I will not necessarily want that to happen all the time. So I need a way of stopping that happening. So what we have in the prokaryotic genes is what we call a repressor molecule. This little black Pac-Man here. And what the repressor molecule does is it sits in between the operator gene and the promoter region, and it stops my RNA polymerase joining, and hence the transcription and the translation happening. Now, obviously, there might be times whereby I want that to transcribe and translate, i.e. if I've got lactose present, okay? So this is the default position. The repressor molecule is in place, but I suddenly introduce lactose. Here's my lactose, this yellow dot here, okay? And my lactose will bind on to the repressor molecule, like so, and then remove that from the operator gene in the promoter region. And this will then mean that the RNA polymerase can join on and cause a transcription and translation, in this case of the lactase enzyme. Now, once the lactase enzyme is produced, it will break down this lactose and take that away, breaking that down into glucose and galactose. So off we go with that. And obviously, at that point, this repressor molecule can join back on, so you no longer produce the enzyme, because you don't need to, because there's no lactose present. Now, that example is called the lac operon and is a prokaryotic example. Let's move on to the eukaryotic situation. So in a eukaryotic piece of DNA, we just have the promoter region. And the default position here is that the promoter region is always open. So the RNA polymerase theoretically could continually bind on, but you don't really want that to happen, or certainly don't want that to cause transcription and translation, because, as we said earlier, there are certain cells that you don't want to produce certain proteins in, and equally, there are certain cells that will only want to produce that protein at certain times. Now, in this situation, simply having the RNA polymerase joined on does not cause transcription. So, if we want to cause a transcription, we need another thing to come along and effectively hold its hand, okay? Now, in this case, I'm going to use the star. And the star will bind on as so, and we're going to call the star a regulator protein. So only once this complex is formed with the RNA polymerase and the regulator protein will the transcription and therefore the translation be able to happen to produce what we call active mRNA. If that's not present, that's not going to work. And if you think about it, what's going to cause the presence of this is, as before, like the environmental stimulus. So for instance, let's say we want to produce, I don't know, insulin, for example. Probably the presence of glucose is going to cause that regulator protein to bind on and complete that complex so that we can get the transcription happening. Hopefully that helps. I've given you a copy of these yourself to use. You can play with these yourself, or you can just drag them around the computer screen if you don't have the printing facilities or a pair of scissors. But hopefully that's useful. This was the lesson that I did for my interview when I got this job, so it must be wonderful. Hope you're all okay at home, and I will be in touch again soon.